Good afternoon. Thanks for inviting me to give this talk at the Protein Metrics Workshop this year. My name is Corey Ralston. I'm a staff scientist at Lawrence Berkeley National Lab at the Molecular Foundry, and I'm excited to tell you about this method called X-ray hydroxyl radical footprinting. So this is a solvent accessibility method that gives you structural information on proteins in solution. So I'll be telling you how we use X-rays to generate modifications on proteins, and then later, Lena Christensen will tell you how we are starting to use the protein metrics software to analyze those modifications. So first I'd like to give you a very brief history of the footprinting method. This has been around quite a while now. So hydroxyl radical footprinting was first discovered in the early 70s, where it was shown that you could use DNA to figure out where protein was binding on a strand of DNA. So the idea behind this method is that DNase will cleave DNA at certain positions, but if the DNA strand is protected by a protein at those positions, then you won't get cleavage. And by comparing the cleavage products with and without protein, you can figure out exactly where the protein was bound. So in the years following that, several other chemical modification methods were developed, and in the late 80s, Tullius's group discovered that you could use hydroxyl radicals to get similar information. Now, the advantage of hydroxyl radicals is that they will cleave the DNA at every single base along the DNA strand. So now if you are looking at a protected region due to some protein binding, and you analyze those cleavage products, you can figure out exactly where that protein was binding. And typically in these experiments, these DNA cleavage products are run out on a gel, and you can watch those protections evolve as the proteins bind to DNA. So in the late 90s, Mark Chance decided to try to use X-rays to generate hydroxyl radicals to see if he could get the same type of structural information as using chemical methods. And he tried this out at Brookhaven National Lab at the National Synchrotron Light Source. And he showed that yes, indeed, he could get the same type of structural information. And the big advantage of using X-rays was that he could get a very high dose of hydroxyl radical generated in a very short amount of time. So that meant that he could start doing kinetics experiments on DNA and RNA on the millisecond timescale. Now, not long after that, he demonstrated that it was also possible to do protein footprinting using X-rays. And protein footprinting is conceptually similar to DNA or RNA footprinting, except that with protein footprinting, we are looking for modifications that are made to protein side chains rather than to cleavage of a DNA or RNA backbone. So here's a movie showing an overview of the method. So we have proteins in solution, we irradiate with X-rays, and the X-rays will generate hydroxyl radicals from water molecules. And if that water molecule happened to have been close to a side chain, the hydroxyl radical will modify that side chain at that position. So we next take our protein and we digest it, and then we run out the peptide fragments on mass spec, and we look for where those modifications occurred. And wherever they occurred, that tells us where the water was around the protein or inside the protein. Now, radiolysis of water is fairly complicated. A water molecule can absorb an X-ray photon and go to an excited state or eject an electron completely, and in either case, radical products will be generated on a very fast time scale. Now these products will recombine with each other and with water to form a steady state concentration of hydroxyl radical and some other products. There are a fair number of radical products generated in this process, but there are two really important things to note about this method. The first is that the hydroxyl radical is the most reactive radical to proteins, so it's causing the modifications that we will see with this method. And the second thing to note is that the radical doesn't diffuse very far before it either reacts with a side chain of a residue or it recombines and effectively disappears. So when we see a modification, we know that there was a water in very close proximity to that side chain. Now in contrast, if the hydroxyl radicals were able to diffuse all over, we would see modifications all over the protein, and this method would not work. So since its invention, X-ray footprinting has been used to look at a number of different protein systems. It's been used to look at ATP-dependent structural changes in some proteins. It's been used to look at photoactivation of some membrane proteins. It's been used to look at transport or gating mechanisms in membrane proteins, how ions are shuttled across a membrane. It's been used to look at the dynamics of waters surrounding and within a protein. It's been used to look at structural changes in ribosome assembly in cells. And most recently, we've been using it to look at the photoactivation of a particular protein and how a pigment molecule moves within that protein and how the domains associate as a function of photoactivation. I wanted to point out this method called fast photochemical oxidation of proteins, or FPOP. In this method, UV laser is used to generate hydroxyl radicals from hydrogen peroxide, and those radicals go on to modify proteins where they are solvent accessible in the same way that X-ray footprinting does. I wanted to tell you in a little bit more detail about how we do these experiments. So typically we collect several different X-ray exposures and we monitor the modification of a residue as a function of that exposure time. So in this example you'll see 0, 10, 20, and 30 milliseconds exposure, and you can see the modification on that residue increasing as a function of X-ray exposure. And we plot the unmodified fraction as a function of X-ray exposure time, and we get a rate. So generally, the higher the rate, or the steeper that line, the more the residue is solvent exposed. And a very shallow line means that that residue is protected from hydroxyl radicals. So in practice, when we do this experiment, we're usually comparing two different states. So for instance, with the protein with and without a ligand. So as the ligand is binding, we're seeing more protections at certain residues, and that gives us information about where that ligand is actually binding. Now, one thing that's kind of important to note about this experiment is that it is a radiation damage experiment, but the dose is actually to the water, not to the protein itself. So we don't see direct X-ray damage to the protein. What we see is hydroxyl radical damage to the protein. So what this means is that we can have a dose, a certain X-ray dose to our solution, and we can see some kind of rate of modification of a residue. If we then change the buffer conditions by adding, say, detergent or glycerol, any kind of radical scavenger, we can be hitting the solution with the same amount of X-rays, and suddenly we see no modification. And so this is because the hydroxyl radical effectively mitigates the X-ray dose to the protein. So because of this, we have to figure out a way to determine the dose to the sample, the dose to the protein. And to do this, we use an empirical measure. We use a dye, an Alexa dye, which fluoresces. But when it's been damaged by X-rays, it stops fluorescing. So if we monitor the change in fluorescence of that dye as a function of X-ray exposure time, that gives us an empirical measure of the amount of dose that the protein will get. So when we do this experiment in practice, before we collect any protein samples, we add dye to our buffer and we collect several X-ray dose experiments to figure out how much X-ray exposure we will need to use when we run the proteins. So one of the reasons I'm telling you this is because these experiments at the beamline can end up being very time intensive, as we have to collect a number of Alexa dose responses before we can actually start with the protein experiments. So this was one of our motivations for developing our inline fluorescence system. So the idea behind this is that we use a jet to eject a sample, either droplets or a stream of liquid, into the X-ray beam. And by controlling the speed of that sample, we can control the X-ray exposure. And we also hit it with a visible light laser as the sample is falling and before it hits the collection tube, and we measure the fluorescence coming off of that sample. And the amount of fluorescence of that dye tells us how much dose that sample has received. And so we can generate this Alexa dose response immediately and then control the velocity as necessary, tune those parameters, and figure out exactly what X-ray exposures we want to use for our protein samples. So this has improved the experiment tremendously and saved us a lot of time at the beamline. This is what the instrument looks like in real life. So we have a syringe pump to push a liquid sample through a nozzle, and a fraction collector so we can collect multiple samples. The X-ray beam hits the sample fairly close to where it's ejected from the nozzle, and the exposure time is determined by the speed of that sample as it passes by the X-ray beam. We have a laser that shoots the falling liquid, and then we collect that fluorescence along the same light path into a photomultiplier tube. From above, this is what it looks like: the syringe pump, the rotating stage, and the jet. And we have here's the laser to excite the fluorescence and the photomultiplier tube to collect that fluorescence. So we were really lucky to have two great students from Sonoma State University who helped us put this instrumentation together and to write a software GUI to control it. So you can see here that the camera image is capturing that fluorescence off of that liquid jet as it's being collected into those collection tubes there. And through the GUI, we can control how many samples to run, how much exposure each will have, how much volume of each sample. And then after the collection, the software will automatically calculate that dose response rate for the Alexa dye. So that automation has helped tremendously with the throughput at the beamline. And as part of Lawrence Berkeley Lab and as part of the Foundry as a user facility, our mission is to help people with science projects, and we would really like to make this method, this X-ray footprinting method, which is a structural biology method, accessible to a lot more people. So we've achieved part of this goal by automating the X-ray part of the experiment. We've developed software for the data collection at the X-ray facility, and now we're working on streamlining the mass spec portion, the mass spec data collection part of the experiment, and on automating the analysis of that mass spec data. So this is our workflow. We do our irradiation at the Advanced Light Source synchrotron using the new automated methods that we have. We then digest and we bring over the samples to the molecular foundry, which is about a 10-minute walk away. We have some mass spectrometry instrumentation there that we would like to upgrade to get to more sensitive instruments, and we are working on that. After the samples are run, then the data will be generated and ported automatically into the protein metrics software, where we're working on automated analysis as much as possible of the X-ray footprinting data. So before I hand it off to Lena to talk about the data analysis, I just wanted to give a big shout out to Sayan Gupta, who has been my partner in crime in developing the X-ray footprinting method at Berkeley Lab and probably understands more about footprinting than I ever will. And these two amazing students, Matt Rossi and Brandon Russell, who helped develop the instrumentation and the software for controlling the experiment. And the mass spectrometry lab at JBay, who helped us develop some of the methods for running the footprinting samples on the LCMS. And Michael Connolly at the Foundry, who's helping us set up some new methods for analyzing footprinting samples. And finally, Lena Christensen, who is the newest member of our group and has been doing an absolutely terrific job learning the protein metrics software and how to apply it to generating dose response curves in the X-ray footprinting experiment.